one of the very important session that we will go through. So I will request Bankar sir again to take the charge of the dais and to start with the second session.
Thank you very much. So let's talk about jurisprudence. प्रभु कौन की चीज रोकते थे? Guidance is like a long way anyway, so that is dissolution jurisprudence. Alright, I am very happy that quite a few of you are back. So either you are giving me respect or you want more to know. Both are in my favor anyway. Okay. So the homework was, during lunchtime, the homework was, can we determine A through B? We know the dosage form A, we are administering it. We know the blood sample that we draw, we can identify how much amount of drug is in the blood. You also know that the drug is administered in solid state in the formulation and the drug appearing in blood is in molecular state. Third, we also know that this molecularization of the drug from the drug in solid state in the formulation cannot happen in blood. It has to happen outside blood. Cannot happen outside body because the drug is already ingested or applied. So somewhere it happens in the body. And that process we said that, well, it cannot be sublimation, it cannot be melting. Third possibility is dissolution in vivo fluids. And I think in vivo fluid at least before 6 o'clock in the evening is always aqueous based. After 6 o'clock the biorelevant medium changes. It becomes colored also. Anyway, that was the sidebar. But so if this process of in vivo dissolution is going to occur with all our deductive logic is happening in vivo. So my question is, can we determine it? Because then we will get closer to in vivo. I will give you an example and then you tell me, is it possible now in anywhere in the world? Cimetidine is a drug which is an old drug from Glaxo, which is for acidity, gastric ulcers. So now it was an NCE at that time and Glaxo wanted to determine A through B and B through C. Why? That time I was a PhD student and I was poor also. Poverty and studentship goes together. They were interested in that and requirement in that because the only way you can designate a new chemical entity as an antacid is on 2 or 3 counts. Number 1, when it is administered orally, the gastric acidity has to come down, which means pH has to go up of gastric medium. Number 2 is that pH has to go above 3 and maintain that for at least 60 to 90 minutes. I am talking in vivo. So that's the reason why it is critical for a NCE, which is an antacid, to determine A through B in vivo dissolution and then determine the pH following administration for next 30 to 90 minutes. So A through B was required. And C, we take blood samples anyway. So what did they do? They had a protocol, clinical protocol, and they invited participation. Of course, substantial amount was paid as compensation. कहाँ साइन करना है, that is more important than क्या देखना है when you are a student, or at least. So essentially what they did was, they said, well, come at 6 o'clock in the evening, then at 8 o'clock you will have dinner, and then you have to sleep overnight for 10 hours, and then morning blood samples protocol was there, and then next day you go back home or wherever. So everything was written in the protocol, but I don't think anybody reads the protocol. They signed it. So essentially, 30 of us, we were there. 8 o'clock, lunch ho gaya, dinner ho gaya. And then they said, okay, what you have to do is, they gave us a balloon, small balloon. On one side of the balloon was a long tube. And on the other side of that balloon is opening. So you have to chew it from the side and swallow. So the balloon went into the stomach with this thing hanging out, the tube. And they said, okay, now you can go to sleep. Morning 6 o'clock, wake up. First thing they did was they took a sample from our gastric medium over fasting. Determined pH, no drug administered, so 0 in vivo dissolution. Took a blood sample. Every 15 minutes for next 45 minutes, they pulled a sample from our gastric medium after administering the tablet, and then they determined the pH of that, and they determined physically how much was dissolved in vivo, A through B. And the blood sample gave A through C. So, that is possible. So after 90 minutes, then fine, they did not take any sample. Regularly whatever the sampling was there, and then 24 hours later, the next morning was the most difficult part. Tube is hanging out, that has to be pulled out. That was painful. That was worth every single dollar that they paid us, or even more it was actually. But A through B was done, was possible. Can you do that today? Today, Ethics Committee and IRB, which is the same thing, will not approve it because it is invasive. Rectal they will approve because you can have a endoscopic camera, but oral they will not approve. So now, I know you didn't know the story, but do you think that A through B is possible? It is not. What we actually see in bioavailability is after the fact, after in vivo dissolution has happened, the result of that. So A through B is not possible. A is possible, I know. C is possible, I know. We have a protocol. So now the question is, how to determine A through B? Why? Why is it important? All changes that you do in your formulation, it will be reflected in dissolution. That is what we determine, or that is what we see here. Hardness change kiya, disintegration time change, dissolution rate changed. Polymer concentration बढ़ाया कम किया, thickness of coat बढ़ाया कम किया, blending of excipients like lubricants and all बढ़ाया कम किया, immediately it will show up there. इन विट्रो, बट इन विवो में क्या करोगे? बंद कीजिए सेल फोन। इन विवो में क्या करोगे? We are hoping that our in vitro dissolution is going to predict this. But we cannot predict A through B in vivo dissolution. We are going straight from in vitro dissolution and determining A through C. आप तो गॉड होने लग गए जी। That's the toughest part. As the drug gets complicated, as the drug solubility changes, as the formulation expectation IR to SR, as the excipients change, as the composition changes, as my batch size changes, as my processability changes, site to site change, we focus on what would happen in A through D, hoping that something similar will happen in A through B. The only thing that we have is the formulation and the blood sample after effects of A through B. So now you can start to realize dissolution का importance कितना significant है. Any guidance that is given, how will that help in development? It will help in targeting your quality control at the end, post-clinical. So if you develop a product, given the guidance never ever says that this is predicting bio. We think it is because FDA ने बोला. FDA does not know your formulation. FDA does not even know your drug. But FDA has a guidance for all types of formulations in that category. So now this test becomes so phenomenally of paramount importance, and if we just follow the guidance, if it is there, without even questioning whether the guidance is applicable for me in the development stage, is a monumental mistake. We are trained to think, we are not trained to follow whatever anybody says, either in public domain or FDA or research article or abstract, blindly. You should not. That is the biggest learning that you should take from this. You take your submission today, which is approved, the batches are being done every day. 1 year later, the product is in existence. Check it whether it will meet bio, which was when you started with. That's how you got approval. Over the time, equipment has been replaced, not changed, replaced. Over the time, your batch size is increasing, has increased. Over the time, because your first 6 months or whatever the X number of batches were done minimum to prove that well, your quality is being maintained, you go for reduced testing. How do you know it will work? Why did FDA have to come up with a rethinking? FDA is the one who said that, well, if I have multiple dose strengths that I am preparing, I can make a standard formulation and then I can dose proportionately increase it or decrease it. If the number of doses are more than 4, now FDA insists, even though your formulation is dose proportionate, you will have to do a you will have to do a confirmatory bioequivalence or biostudy of the intermediate dose. Why? ये डिसोल्यूशन जो है वो लगता है सिंपल है, क्या है यार, ले लो टैबलेट डालो, सैंपल ले लो, जाके चाय पियो, आपको एनालाइज करो, हो जाएगा. This is where your expertise should come into place, or your thinking and your rationalizing has to come into place, which will bring in physical chemistry, which will bring in chemistry, which will bring in biochemistry, which will bring in physiology. All come together where A through C is controlled only by your body, physiological. It is a one-way traffic. A through D is a multi-way traffic. So what could pass in vitro may not pass A through C. Not predictive. क्यों? A through C is we did not think about actually what should be done, which is closer to it, to the in vivo condition A through B. Albendazole को मैंने लंच के टाइम पे पहले बोला, poorly soluble drug, 20% of the dose is dissolved and gets bioavailable. We are doing A through C for 100% dissolution, 85% guidance है. If you do it in the right environment in A through D, which is coming A through D, which is coming close through A to B, जितना आप नजदीक जा सकते हो उतना. You cannot do the balloon study anyway, so try to get as close as possible. And then, honestly, with only 40%, given abundance of caution, even 40% of the drug dissolved in that particular condition, appropriately sampled, your chances of getting A through C is much higher than blindly 85% करो क्योंकि guidance मिलेगा. I'll give you a classic situation and then this should kind of make the register in your mind. रेक्टल सपोजिटरी के लिए इन विट्रो डिसोल्यूशन टेस्ट रिक्वायरमेंट 0.1 नॉर्मल HCl कर सकते हो क्या? Point 1 normal HCl is acidic, correct? pH 1.2. रेक्टल सपोजिटरी है एंड डिसोल्यूशन टेस्टिंग सॉलिड डोसेज फॉर्म के लिए करना चाहिए. There are a couple of guidances which say that dissolution of a rectal suppository should be done at 1.2 pH. किसका रेक्टम इतना एसिडिक होगा? विंडलो, चेट्टीनाड, जो भी है, कोल्हापुरी, लाल वड़े और when you look at the food, your mouth waters, when you eat it, your eyes water, and the next day is a problem. Suppository के लिए 1.2 pH लगा रहे हैं dissolution को. So what is the relevance? और उसमें हम प्रेडिक्ट करने वाले बायोअवेलेबिलिटी रेक्टम. So this should start you to think कि भाई what my drug is, where my target is, what I am administering, and what is the in vivo closest where the drug will dissolve and get absorbed. That will change for every drug, that will change for every formulation, that will change for every process. So now when you start to come together, bring this all together, फिर ये मॉडल एकदम लगता है, हाँ इसमें कोई दम है. let's take it forward. A is the drug in solid state, which is going to be, we want it to release in vitro or in vivo going to A through B or A through D, doesn't matter. So particular stage, let's say it is highly disintegrating, rapidly disintegrating, immediate release formulation. But the drug is poorly soluble. तो पहले क्या होगा? Disintegrate हो जाएगा, drug के particles आ जाएंगे बाहर. Then they will dissolve, correct? Yes or no? Yes. So A through B requires that not only the drug should be released and then it will dissolve. दो steps है. दो steps है. Okay. More important part is ultimately the particle has to dissolve. किसमें? Biological fluid में B. Ocular है तो lacrimal fluid. ओटिक है तो कान का fluid. Dermal है तो dermal fluid. Rectal है तो don't answer the question. You have to have a simulated part. Angal hai, you have fungal infection underneath the skin. So we have medicated lips, nail polish. So the drug has to pass through, so you need to have an understanding of what fluid will be under the nails, which is different. Injectable, subdermal, transdermal, intradermal. So this is where now our understanding starts, but no matter what, the particle or so-called the drug has to dissolve or dissolve in the medium which is aqueous. Uske ionization or so-called ionic concentration and pHs might be different from different locations where it is dissolving. But जो भी है वो fluid is aqueous. अगर उसमें उसकी solubility है ही नहीं तो dissolution क्या खाक करोगे? I have a practically insoluble drug. Most of our drugs are weak acids and weak bases, hydrocarbon compounds. So now, first thing we have to do is कि भाई इसमें soluble है कि नहीं. उसके बाद में देखेंगे formulation से निकलने के बाद dissolve होता है कि नहीं. So the first requirement is solubility. Second requirement is dissolution. If you don't have solubility, you will not get dissolution. You may still get bioavailability because weak acids and weak bases. आपका physical chemistry मार्टिन का बुक ले लो, आयनाइजेशन का चैप्टर 6 या 11 है जो भी एडिशन पे पढ़ लो, उसमें दिखाना है कि when it dissolves or when it solubilizes, first it has to get into the medium. If that is not possible in aqueous medium, or it is possible, it has to happen. Dissolution is the second step. First is solubility. Can you differentiate between solubility and dissolution?
Cmax.
Bioavailability नहीं पूछ रहा हूं मैं. Solubilization prerequisite for dissolution. हम दोनों एक साथ ही बोलते हैं. Solubilization is take the pure drug and put it into all media of interest. आपने फिजिकल फार्मेसी में एक्सपेरिमेंट सॉल्युबलाइजेशन का किया है, उसमें आपने K sub S सॉल्युबिलिटी डिटरमिन की हुई है. लगाओ जरा दिमाग.
solubilization में forcefully we can include the changing the factors like temperature and that, and dissolution we normally
secondary पहले solubility होना चाहिए, then I can do a lot of stuff. So what we do is we take unknown amount of drug, doesn't matter, you take a test tube 25 ml या 10 ml ले लो, whatever solvent you want of interest, उसमें ज़्यादा डाल दो, shake it for overnight and let it settle. Next morning you take a sample from the supernatant, which is saturated amount of drug in the solvent, that is solubility. That has to happen. Pure drug in a solvent. Now this drug of interest, for 30 milligram dose, you are putting it into a composition. Now it has to come out. Oh, disintegration aa gaya drug bahar. Now it will solubilize. But that is dissolution coming from the formulation. So whenever you are talking about dissolution, it is the solubilization of the drug post release from a formulation. पर पहला solubilization होना चाहिए, उसके बाद ही dissolution हो जाए, इसलिए all of our activity is apparent dissolution. There is nothing like absolute dissolution, जो solubility में constant है. Okay? Now, interesting part. I have got a highly soluble drug, and the product is rapidly disintegrating and dissolving, such that 85% of the dose is dissolved in less than 15 minutes. तो ये दोनों स्टेप में first solubilization, then dissolution. Solubilization के लिए ड्रग बाहर आना चाहिए, उसको हम रिलीज बोलते हैं. Both these steps are time dependent. Time dependent means there is a rate. 30 मिलीग्राम ड्रग रिलीज होने के लिए कितना टाइम लगता है, एंड फ्रॉम फॉर्मुलेशन से रिलीज होने के बाद डिसॉल्व होने के लिए कितना टाइम लगता है. If the release of the drug is more rapid, the only thing that is required is then dissolution, which is slow, because इतना highly soluble drug है, rapidly disintegrating है, so the release is pretty quick, but the dissolution is slow. तो दो rates में dissolution is slow, release is rapid. तो rate limiting step कौन सी हो गई? Dissolution. Hello? Low, जो slow होता है उसको rate limiting होता है. You know that from our stability and testing, first order reaction, second order reaction, हमने सब क्या हुआ है वो. Okay? So now these drugs, where the drug release is rapid but dissolution is slow, they are dissolution controlled. वही ड्रग हम सस्टेन्ड रिलीज पे डालते हैं. Now I make sure that the release is slow. आपके सब fantastic technology, coating and micro encapsulation and double coating and ये और वो सब. Where I ensure that the release is slow, but once it releases, it dissolves pretty quickly. So now what is the rate limiting step? Release or dissolution? Release. दो टाइप्स ऑफ फॉर्मुलेशंस आ गए. कंपेंडियल मेथड्स में तो 711, USP 711 and 724. 711 is entitled dissolution testing. 724 is entitled drug release testing. Method 1 and method 2 कॉमन है. USP को क्या दिमाग खराब हो गया? सेम इक्विपमेंट है, सब सेम है, तो अभी ये फर्क क्यों आ गया? When you read that small print USB में, तभी वहां लिखा हुआ है dissolution 711 is preferred using method 1 and 2 for rapidly dissolving formulations, immediate release formulations. The moment you read 724, it says for sustained release products, extended release products, modified release products, because it is release rate controlled. तो रिलीज और डिसोल्यूशन में फर्क है. अगर आपको यहां से जालना जाना है और बोला कि भाई जालना यहां से जाना है, तो आपको सिर्फ आधे घंटे देता हूं. जो भी फैक्टर्स इन्फ्लुएंस हो जाएंगे, स्पीड, आपका पेट्रोल है कि नहीं, स्पीड, बाकी का वेदर, बाकी किसको हाई बोलना है, बाय बोलना है, वो सब आधे घंटे में करना है. So the influence of factors in terms of getting to Jalna in such a short time are going to be less affecting the result. लेकिन अभी बोला कि यहाँ से जालना आपको 3 घंटे देता हूँ, तो आप बैठ के बाजू में उडुपी का चाय कॉफी लेंगे, फिर वहाँ जाके डोसा लेंगे, फिर ओ हाय गर्लफ्रेंड, हाय बॉयफ्रेंड करके मिला, कल थोड़ा गॉसिप भी करेंगे, फिर भी 3 घंटे में पहुंच जाएंगे. ऑल फैक्टर्स अफेक्ट मोर एज इट इज बिकम लॉन्गर एंड लॉन्गर. सो इमीडिएट रिलीज फॉर्मुलेशंस, जहां डिसोल्यूशन टेस्टिंग इज द डिसोल्यूशन स्टेप इज द रेट लिमिटिंग स्टेप, तो हम कैसे इवैल्यूएट करेंगे फॉर इन्फ्लुएंस ऑफ फॉर्मुलेशन डिफरेंस? डिसइंटीग्रेशन टेस्ट को हम इतना लगा देते हैं कि ओ थोड़ा डिसोल्यूशन स्लो हो रहा है, तो चलो सुपर डिसइंटीग्रेट आएंगे. अगर वो नहीं हुआ तो एजिटेशन रेट इतना घुमा देंगे कि बाहर आना ही चाहिए 20 सेकंड में. अंदर एक ही सिस्टम है जी, जहां फॉर्मुलेशन के कैरेक्टरिस्टिक्स आर गोइंग टू बी मोस्ट क्रिटिकल, वो बाहर आना चाहिए, बस जाएगा अंदर, स्लोअर. So now you tell me which factors you will concentrate on during development of an IR product where dissolution is the rate limiting step, not release. Sustained release factor में exactly I apply the same thing and say now you will see all biological factors. आराम से वेकेशन टाइम है जी, 2-2 घंटा, 12-12 घंटा, 24-24 घंटा लग रहा है आपको. So now you tell me A through B is going to be more affected or influenced in a formulation that is modified release or SR, और वो हमको मिमिक करना है A through D में to predict A through C. समझ आ रहा है? गाइडेंस से अब नहीं मिलेगा आपको कुछ. एक मॉडल है बस. तो A through B is our real catch point. In order to really check what will be our A through C. B through C तो एक ही वन वे ही है, फिजियोलॉजिकल कंट्रोल. नाना पटेल बोलता है कंट्रोल, फिजियोलॉजिकल कंट्रोल. सो अगर हम बायो स्टडी कर रहे हैं नेचुरल सब्जेक्ट, नॉर्मल सब्जेक्ट्स में, या 21 डिफरेंट प्रोटोकॉल रिक्वायरमेंट्स टू इंक्लूड और एक्सक्लूड अ सब्जेक्ट, उसमें हम A through B का environment we try to control, and then hopefully B through C will be relatively the same in the normal subjects. A through D में तो क्या करते हैं, वेसल तो वेसल ही है, और क्या कंट्रोल करोगे? मैक्सिमम एनवायरमेंट इन द वेसल, pH करो, ये करो, वो करो, with the surfactant, without the surfactant, with the sinker, without the sinker, to be type of mixing, type of agitation, वो सब. B through C को कुछ फर्क नहीं पड़ता है, सब फर्क A through B करते हैं, पड़ता है. एंड हमारा पता नहीं पिछले कितने जन्मों का बुरा कर्म है कि हमको A through B पता ही नहीं सकता, मिली नहीं, कैरेक्टराइज नहीं कर सकते हो. लेकिन A through D को हम कर सकते हैं. Anticipating what is in A through B. Without knowing your physiology, without knowing your biological requirements, without knowing your biopharmaceutics, कैसे डेवलप करोगे आप डिसोल्यूशन टेस्ट? फिर राम भरोसे, तुक्का लग जाएगा तो अच्छा है, नहीं तो करते रहो, लगे रहो मुन्ना में डिसोल्यूशन, क्या करो, डेवलप करो. So all of our efforts ultimately come down to understanding of A through C, to get close through A through B, which we cannot determine actually, hoping that B through C will always give us expected outcome. So hamara all the activity is in this area, saying that we want to predict this. Oh, wow, हम तो very great हो गए. इसके ऊपर हमारा कंट्रोल नहीं है, इसके ऊपर कंट्रोल है, पर we cannot determine it. So unless you know where, how, when, why, then using this to predict this is a worthless activity. Absolute worthless activity. तो ये थोड़ा 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 करके आ जाएगा. I am explaining it to you in simple way. It appears like, oh, बहुत काम है, बहुत काम है, बहुत काम है. नहीं. You know the drug, you know the endpoint, you know the physiological requirements. Now you start to think about, okay, ऐसा करना है तो क्या करेंगे? So at least we know if I am giving it orally, I want this medium to be gastrointestinal tract medium. So I simulate A through D का medium close to gastrointestinal tract. But my project is in ophthalmic delivery with formulation to be placed in the lower eyelid, lip, where film है वो. कैसे करोगे? What fluid? Lacrimal fluid. तो कल डिसोल्यूशन करना है तो आज रात को देवदास मूवी 4-5 बार देखो, कल सवेरे जाके रो लो, लैक्रिमल फ्लूइड डिसोल्यूशन करो उसमें. So now we start to see the need for an appropriate medium for A through D, which will get close through A through B. लेकिन हम close पर वैसे नहीं होगा ना. She is almost like the one I have idealized, but she is not that. Same thing with he also. Huh? Mother-in-law हमेशा बोलती है, she is like my daughter, but not my daughter. माझ्या सुनेत मी माझी मुलगी दिसते पण मुलगी नाही. So that's where our problem starts as to, well, unless we know what we want in A through B, this whole exercise of A through D becomes futile. Number two, now we want to know how this A through D happens. Without knowing that, again it is worthless. Now put it together, we not only need to know what it is required, how to do this, but we have to also know when and which way. फिर भी हम A through B तक कभी भी नहीं कर सकेंगे 100% रिप्रोडक्शन, कभी नहीं कर सकेंगे. See now how much responsibility we are putting on the dissolution test. तो dissolution test के तो पर ही लग गए, which was simple a quality control test. Now this has become the monumental test. Dissolution lab में जब चालू करोगे डेवलपमेंट, हर एक सवेरे जाके पहले उसका पूजा करोगे, बयास कर दो कुछ नहीं होगा, क्योंकि we have to know what to put in there to begin with. Okay, all right. So now A through B, first step is we need to know solubility. Then we also need to know what is dissolution. Simple difference, ध्यान में रखना है, dissolution is apparent from the formulation. Solubilization is the pure drug alone. No excipients, no process, nothing. अगर मैंने आपका प्योर ड्रग का एक फॉर्मुलेशन बनाया जिसमें कोई भी एक्सिपिएंट नहीं है, सिर्फ ओनली एक स्टेप है, मुझे डिसोल्यूशन उसका देखना है. सो व्हाट आई विल डू इज आई विल रिमूव any excipients, कुछ भी factors नहीं लाऊंगा, except that I need some kind of an image of my product, so I will just take the drug and compress it. Only एक factor आ गया, compression, and I will further restrict it to dissolution of the drug from this so-called compressed matrix or a pellet, का सिर्फ एक surface expose करूँगा to that medium of whatever I want to evaluate in. And I will know the surface area because मुझे पता है उसका dimension क्या है. Everything I have controlled other than just one step of formulation, which is just compression. So that will be the intrinsic dissolution, क्योंकि उसमें formulation है. So now intermediate step is पहले drug का solubilization करो, then you do its intrinsic dissolution, फिर आपका जो formulation के तारे तोड़ने वो तोड़ो, make your formulations, and now you see the apparent dissolution of the drug. IR है, MR है, pulse release है, whatever it is. That is your technology. So ये progression है. टेस्ट करोगे तो कैसे डिटरमिन करोगे इसका मीनिंग, कैसे निकाले वो डेटा से. सो वी लुक एट टू थिंग्स. दिस टेस्ट डिटरमिन्स द अमाउंट ऑफ टाइम एंड एक्सटेंट ऑफ डिसोल्यूशन. सो अमाउंट डिसॉल्व्ड ओवर टाइम इज रेट एंड टोटल अमाउंट डिसॉल्व्ड इज एक्सटेंट. ये इंपॉर्टेंट है, नॉट बिकॉज हमको मैथमेटिक्स आती है इसलिए. बायोअवेलेबिलिटी का डेफिनेशन का क्या है, रट्टा मारा है आपने बायोफार्मास्यूटिकल क्लास में. बायोअवेलेबिलिटी इज डिफाइंड एज रेट एंड एक्सटेंट ऑफ द ड्रग इन सिस्टेमिक सर्कुलेशन फॉलोइंग एडमिनिस्ट्रेशन ऑफ अ यूनिट डोज, यूनिट मास व्हिच इज डोज, अंडर स्टैंडर्ड कंडीशन व्हिच इज प्रोटोकॉल. वहां रेट और एक्सटेंट चाहिए ए थ्रू सी में. That's the reason why we want to concentrate on A through D, on rate and extent. लेकिन हमारे गाइडेंस क्या बोलते हैं? Immediate release formulation. Somebody is really into it. Good. Guidance क्या बोलते हैं? Immediate release formulation, rapidly dissolving drug, highly soluble. 85% dissolved, cumulative in 45 minutes, 30 minutes. So it is talking about extent. What about rate? Guidance किसने बोलता है? क्यों चाहिए? क्यों? लेटर क्यों चाहिए नहीं? All dissolution guidances are Q, determination of percent dissolved cumulatively at whatever time. But A through B is a rate process, B through C is a rate process, A through D is a rate process. Bioavailability A through C, we want it in terms of rate and extent. और डिसोल्यूशन तो सिर्फ एक्सटेंट बोल रहा है. When I go from an immediate release formulation to a extended release formulation, क्या फर्क है? रेट में फर्क है कि एक्सटेंट में फर्क है? $60 मिलीग्राम डेल्टा एसएम हाइड्रोक्लोराइड का इमीडिएट रिलीज कैप्सूल है. 60 milligrams of diltiazem hydrochloride, same drug, which is also twice a day, 12 hours. तो उसमें फर्क क्या हो गया? रेट में फर्क है. एक इमीडिएट रिलीज फॉर्मुलेशन विच इज रैपिड रेट, दूसरे में सस्टेन्ड रिलीज फॉर्मुलेशन विच इज रेट, स्लो हो गया. बायोअवेलेबिलिटी भी हमारी स्लो हो गई रेट में. एक्सटेंट में फर्क पड़ा कुछ? यस और नो? 60 मिलीग्राम का 60 मिलीग्राम से आएगा. 30 मिनट्स में आए या 12 आवर्स में आए? आया और ऐसा इट इज ओनली 60 मिलीग्राम. लेकिन कैसे आया? फास्ट आया कि स्लो आया? रेट. That is the distinguishing factor between IR and sustained release or modified release. Rate, not extent. All my guidances are in extent. अगर रेट चेंज हो गया तो हमको पता है कि एक्सटेंट तो नहीं चेंज होगा. डोज तो डोज ही है. So now you tell me the parameter which is more sensitive, rate or extent. स्लो करते जाओगे, डोज सेम है तो रेट में फर्क पड़ेगा ना, एक्सटेंट में नहीं पड़ेगा. All your formulation development is controlling the rate, but you are following a direction which is an extent. So chances of failure तो इधर ही दिख रहे हैं मुझे right away. Two trains will reach the same destination, but one is going slow and one is going rapid. Destination तो वही पहुंचेगी. बिहार में लालू प्रसाद was the railway minister. तो he was interested in कि गाड़ी सब right time होनी चाहिए. ठीक है भाई. तो एक बार पता चला कि ये ट्रेन आ रही है ऑन टाइम. वाह. तो स्टेशन मास्टर ने कहा कि भाई आने के बाद जो ड्राइवर है उसको उसका जरा ये करो, फेलिसिटेट द ड्राइवर, उसको गार्डन बिल्डर ला के राइट टाइम लेके आए. तो आ गई गाड़ी ऑन टाइम. तो गार्डन बिल्डर डालने लग गए. तो ड्राइवर थोड़ा चकमा आया कि भाई क्या चल क्या रहा है. तो उन्होंने कहा सर आपने गाड़ी राइट टाइम लेके आए. अच्छा रियली? 24 घंटा लेट है जी. रेट में फर्क पड़ा ना, गाड़ी तो पहुंच गई. whenever you are doing, you should be concentrating on rate during development. When you are doing quality control, you should be concentrating on extent, because all quality control is to be reported in Q, not dQ by dt. अभी पता चला क्यों। बोलने में तो इजीली बोल देते, अरे उसको थोड़ा स्लो कर दो यार, दैट मीन्स रेट। कम डोज डालो नहीं बोलेंगे, सीमैक्स अगर फेल हो गया तो कम डोज डालो नहीं बोलेंगे, थोड़ा उसको स्लो कर दो या फास्ट कर दो। So you are dealing with rates, but you don't acknowledge that, क्योंकि हमारा mentality, we are not trained for that. क्यों? Because guidance लिखा तो guidance हो। So once you know that guidance is not for bioavailability and bioavailability is all rate, so if you concentrate on rate, automatically you will get the extent anyway, because dose is not changing. So two formulations having the same dose, if their extent is the same, they may have a different rate. एक दिन लेट गाड़ी पहुंची। But two formulations, if have the same rate, वही टाइम पहुंचेगी, they will have the same extent. तो बायोइक्विवेलेंस में फेल होने का पहला दिमाग में आना चाहिए कि भाई हमको रेट कंट्रोल करना चाहिए। सेकंड आना चाहिए कहाँ रेट कंट्रोल करना चाहिए। एक्सटेंट की बात करने की जरूरत नहीं है, क्योंकि वो तो चेंज होने वाला नहीं है। लेकिन हमारा ऑल एक्शन स्पीक ऑपोजिट ऑफ व्हाट वी डू। क्यों मोसेस जी ने एमडीए से आके कहा भगवान ने कि क्यों करो, क्यूमुलेटिव करो, पर मोसेस ने कभी नहीं कहा कि ये बायोएवेलेबिलिटी प्रेडिक्शन है, ये लिए यूजफुल है, हमने लगा दिया उसको। कंफर्ट जोन, फीलिंग गुड। ऑलराइट, when we are doing development, we should determine rate and move forward. That is prospective. 12 घंटे में मुझे ये डोज डिसोल्यूशन होना चाहिए, प्रोस्पेक्टिव। अभी मेरा हो चुका है, लेकिन मुझे बैचेस जो मैंने 10 बनाए हैं उसका रेट सेम है क्या, दैट इज रिट्रोस्पेक्टिव क्वालिटी कंट्रोल, बैच टू बैच वेरिएबिलिटी। तो ये टेस्ट दोनों जगह यूज कर सकते हैं। कोई भी ड्रग हो, कोई भी फॉर्मुलेशन हो, B1 क्लास 1 2 3 4 2A 2B जो भी है, उसका जब डिसोल्यूशन करोगे वॉट एवर टाइप ऑफ फॉर्मुलेशन, उसमें अल्टीमेटली ये 3 फैक्टर्स आर मोस्ट क्रिटिकल। कितने हम अभी वेरी क्विकली देखेंगे। अगर मुझे 60 मिलीग्राम ड्रग डिसॉल्व करना है एट इन अ पर्टिकुलर मीडियम व्हिच इज एक्वियस, उसमें 60 के 60 में जरा अंतर तो जाना चाहिए। क्यों? Why do we need all of the drug dose in the formulation to dissolve? Regardless of whether it is highly soluble drug or poorly soluble drug. Why? क्यों? FDA ने कहा है। Don't give me that. A through B dissolution होता है, हम डिडक्ट करते हैं। ब्लड सैंपल में आ गया मीन्स A through B हो गया। कितना हो गया पता नहीं, कैसे हो गया पता नहीं, लेकिन हुआ, नहीं तो ब्लड में आएगा नहीं। कितना हुआ डोज का पता नहीं। So we don't know how much of the dose in vivo when we administer the drug will dissolve. Of the dose that is dissolved, how much of it actually got into blood, we don't know. हमको सिर्फ पता है ब्लड में ड्रग आ गया मीन्स इन A through B हो गया। कितना, कब, कैसे, आई डोंट नो। लेकिन अब इन विट्रो में मॉडल लगा जा रहे हैं। डिसोल्यूशन टेस्ट नया प्रोडक्ट करो तो पहले दिमाग में क्या आता है लोगों का इंडिया में, कौन सा मॉडल फिट होगा, हिक्सन कोवेल या हिगुची मॉडल? तो सबको तो जान से प्यारा है, आई डोंट नो व्हाई। कभी ओरिजिनल पेपर पढ़ा है हिगुची का? मेरा भी मॉडल है, लगाओ पढ़ो, लेकिन मेरा पहला पेपर नहीं। इनवर्स ऑफ डिसोल्यूशन रेट एज अ फंक्शन ऑफ स्क्वायर रूट ऑफ टाइम, स्ट्रेट लाइन आ गई R.999 बोलेगा तो मेरा हो गया, जिंदगी डिसोल्यूशन की बन गई। क्या जरूरत है? ना हमको पता है कि A थ्रू B में कितना डिजॉल्व हो रहा है, ना हमको पता है जो डिजॉल्व हो गया उसमें से कितना ब्लड में आ गया, लेकिन ब्लड में आ गया तो पता है कि डिजॉल्व हो गया, दैट इज करेक्ट। द फर्स्ट टू वी डोंट नो। अगर वो नहीं पता तो हमको डिसोल्यूशन में इन विट्रो में कितना चाहिए हमको कैसे पता चलेगा? क्वेश्चन समझ आ रहा है? A थ्रू B हमको पता नहीं कैसे करना है, पता नहीं कैसा होता है, लेकिन आई नो A थ्रू B इज रिक्वायर्ड टू गेट थ्रू B थ्रू C, क्योंकि ड्रग ब्लड में आ चुका है। तो A थ्रू B has happened and B through C has happened. I know A through B is influenced by the formulation, वो पता है, लेकिन I cannot determine। और जो A through B हो गया है, जो I don't know how much, उसमें से कितना ब्लड में आएगा वो भी I don't know, पर ब्लड में आ चुका है। तो हमको प्रेडिक्ट करना है इन व्यू एडमिनिस्ट्रेशन करने के बाद जो ब्लड में आता है, आई हैव टू प्रेडिक्ट फ्रॉम इन विट्रो डिसोल्यूशन A थ्रू D, जबकि हमको पता ही नहीं कितना है। तो क्यों लगे हो 85%, 90%, 100%? और उसमें और हमने लगा दिया कि भाई ultimately dose is known, so rate is important. Because we do not know how much A through B has happened, and we do not know how much of the A through B gets into blood C. The only thing we know is blood level में 0 to infinity। 0 में ड्रग आ गया, सैंपलिंग करने के टाइम ऊपर चला गया, नीचे आ गया, and then there is no more drug in blood. So my bioavailability according to definition is all the time when the drug is in blood, right from time 0 administered to the time when there is no drug in blood. That's all I know. अगर वो सब कलेक्ट करोगे तो टोटल अमाउंट ऑफ ड्रग इन मल्टीप्लाई बाय 6 लीटर्स बिकॉज ऑफ ब्लड, अगर वो कलेक्ट करके टाइम करोगे तो आपको पता चलेगा कि टोटल अमाउंट ऑफ ड्रग इन ब्लड इज दिस एक्सटेंट। एक क्वेश्चन पूछता, चलो देखते हैं आपका, अगर मैंने 100 मिलीग्राम ड्रग लिया, इंजेक्शन बनाया, स्ट्रेट ब्लड में डाल दिया C, तो ओनली थिंग दैट कैन हैपन इज द ड्रग विल गेट एलिमिनेटेड, कॉन्सेंट्रेशंस कम होते जाएंगे ब्लड में विद टाइम, हाफ लाइफ में आपको पता है। सो आफ्टर 4 हाफ लाइफ्स ऑलमोस्ट देयर विल बी नो ड्रग इन ब्लड। 0 टाइम को 100% डोज इज इन ड्रग, सॉरी इन ब्लड, बिकॉज मैंने इंट्रावीनस ही दे दिया सीधा C ब्लड। 4 हाफ लाइफ के बाद लेट्स से द हाफ लाइफ ऑफ द ड्रग इज 8 आवर्स, 32 आवर्स में कोई भी ड्रग ब्लड में नहीं है। तो ये जो सैंपल्स आपने 32 आवर्स कलेक्ट करोगे, वो सब ऐड करके उसको मल्टीप्लाई बाय 6000 ml ऑफ ब्लड,